Okay, it's the hour, so I think we can start to at least introduce our presenters. So good morning, good afternoon, everyone. Welcome to our eighth session now for this webinar. So as always, please note that you are automatically muted, but as soon as you raise your hand or if you need to speak, you can write in the chat and I can give you permission to unmute. You are free to write in the chat and in the Q&A anytime, and we will take all the questions at the end of the meeting so that the presenters can respond to you. As a reminder, all presentations will be shared and all recordings will be shared after the webinar series has ended. So that is after the 12th of September. So with that, I see there's 32 participants connected already, and I'm sure more will trickle in as the time goes. So Marie-Noëlle, I leave you the floor.
Okay, thank you, Alexandra. Good morning, good afternoon, good evening, everyone, wherever you are, and welcome to today's webinar on the basics of bioinformatics. So we are very excited every day to have such a diverse group of learners and professionals who are joining us. So in today's session, we will explore the foundation of bioinformatics, its importance in today's scientific world, and the tools and the technique that drive the discovery in the life science. So we have the honor I have the honor to introduce Dr. Mathieu Rouard, who will make the presentation. This morning we have two presentations, the first one by Dr. Mathieu Rouard. Mathieu, you have the floor. Thank you.
Thank you, Marie-Noëlle. Let me first share my presentation. And should be.
Yeah,
we can see it. Okay. Thank you. So good morning, good afternoon, good evening, everyone. So my name is Mathieu Rouard. So I work for the Alliance of Bioversity and CIAT. My expertise, my background is in genomics, bioinformatics, and data management. And I have been working for more than 20 years now in the banana program, where is the international collection in banana. And today I'm going to talk about data management in genotyping. So last week, we already had a nice presentation, and Carolina already listed a number of genotyping platforms. And it's clear for now that there is a lot of platforms that are generating a lot of sequencing data. So those sequencing data are very useful for many use cases, but we also have to acknowledge that managing the data, the sequencing data, can be demanding. So there is a need for appropriate storage capacities, analytical tools, computing power, and this is what this presentation is going to talk about. So if we take some example in the CGIAR of projects that generated genotyping data at large scale, let's say that we have several, but there are three main ones that are quite iconic, I would say. It started with Erie and the 3000 RISE genomes some years ago, where 3,000 accessions from the Genebanks were fully resequenced, leading to a lot of data. And those data were managed also in a database that is called SNP-seq, where you can access those data set. It was followed also by large genotyping sequencing for the WIT accession from the CIMMYT collection. Similarly, CIMMYT had also to deal with the data and put online a Germinate platform to give access to this data set, at least for download. And more recently, there has been also a project, a pan-genome project on chickpea that led to the full sequencing of more than 3,000 genomes of chickpea. And similarly, again, a database was put in place at CGIAR, which is called César. So those databases are actually what you can see from the web, how you can access the data, download the data. But managing data is more than that. It's more than putting the data online. You have a full life cycle for research data. And it starts with number one from the beginning with a good data management plan. This is where you are going to identify the type of data you are going to generate, where it will be stored, who will be managing the data. So a lot of elements have to be captured in a good data management plan for any projects. Then you start collecting the data. So it relates to the sampling, which sampling I'm going to use to create this data set. What type of permission do you have for the sampling? Is it coming from the Genebank? Is it coming from collecting mission? So you have to set up all those information in the collecting stage. And this is where a lot of attention has to be put into metadata management to capture this information from the start, and also to decide what will be the standards you will be using to annotate your data. Dealing with material from Genebank, obviously passport data, good passport data need to be captured, but if you are doing phenotyping, you have other standards in ontologies or like CIMMYT that could be used. Then you receive the data and you need to process them. And the first step will be about looking at quality control. You will have to decide which pipeline you are going to use, which software to produce additional data. Those pipelines usually are going to convert different files into different formats. So you have to track all those steps in this processing step, which is also very important. So at this stage, let's say that data has been generated automatically by the processing stage, and you will start the analysis. And this is where you have more human intervention in terms of file curation, creating a lot of intermediate files up to the final file for your analysis, and where you have to pay about controlling the version. I think we have seen a lot of Excel files, sometimes called final, final 1, final 2. So this is what we are talking about versioning, making sure that you identify the proper files that are going to be archived, which is the next step, the archiving, because those files can be big. So you need to have a solution for backup, for archive. You need to make sure that the files are not corrupted, that you need to take care about data integrity. And only after this step, we can think about sharing data. Which online repository I'm going to select. Then you have to get back to your founder compliance, if it is in the framework of a grant, or what are the legal compliance you need to respect for your material from your sampling and the permission that you have collected at the beginning. And only when everything is done like that, we can think about using the data, looking at the citation, reuse of data, and possible collaboration. So, what type of data are we talking about when we discuss about genotyping data? So we have different types. Obviously, from the sequencing machine, you have the raw read data that arrive in different formats. They are usually very big files, so hundreds of gigabytes, terabytes of data. Then we have the metadata, what I described in the collecting stage. So this data is small but important, text file, usually a tabulated file with different standards that can be used. Then after the processing stage, usually you have also variant files. So they can be stored in different formats. The main one is the VCF, the variant calling format, but you have different ones like AppMap. Dartseek is using a specific one on tabulated files. So those files are a bit smaller than the raw reads. And for some projects, like the one I described before, you can also have some genome assembly. So some assembly, some gene annotation, and that are usually converted into FASTA and GFF3 formats. So for the presentation, for now, I'm going to focus on those two types, the metadata and the variant. And for that, in CGIAR, we have been exploring a tool that is called GIGWA. And I would like to give you more information about it in the next slides. So, as you must know, there are multiple international collections around the world in CGIAR. They all create genotyping data, sometimes with different technologies, different scale, but they all have data to manage. And it has been identified that data management was one of the bottlenecks. And that's why we started a project a few years ago to put in place a tool that is called GIGWA, which has the advantage to be open source, easy to use. It is modular, interoperable, quite scalable, so it can deal with big data sets. and is free. It's a free software to use. So in more detail, GIGWA can store large amount of genotyping data, so up to billion of data, billion of marker. So it's an online system. We say it's user-friendly because it's an interactive system like a website. You don't have to be a bioinformatician. You just need to go through the graphical interface. And through this graphical interface, you can share your data, you can explore your data, visualize, filter it, and re-export some subset of data in multiple formats. So if you attended this webinar last week, my colleague Julie Sardos gave some insight about the VCF file, how the data are stored from the genotyping sequencing. So this format is accepted by the software, and GIGWA can accept it, but it can also work with other type of format. It works with HapMap, it can work with PLink, Flapjack, and also Dartsec natively, so you can import your file in the database. Okay, sorry, I mute myself. Once it is done, you can see this type of interface, which is a typical GIGWA interface that is subdivided in different parts. So your data are first in a database that can be structured in multiple projects. And for every project, you can list all your markers that were in the file we mentioned before. And for every individual marker, you can list all the individual and their different genotypes with all additional information and metadata that is captured by a VCF file, for instance. So you have also the option to, beyond exploring, you can compute some statistics about your file, looking at variant distribution, looking at FST. You can filter, which is very important. You can filter your marker by different chromosomes. You can look at additional filtering options like the minimum allele frequency, the MAF, the level of missing data. If your data are annotated with genes, you can also filter on gene location. Also quality data, RIP depth, all this information that may be available in your VCF file or other format are automatically captured in the database, and you can interact directly in real time with the interface to filter your data. And you can also create some groups, and you can discriminate your marker between groups. So let's say that you have one group of wild material and cultivated material. You could be interested to see what are the SNPs that are discriminating those two ones. And this can be done automatically by the system. And so that's how you can do it in the interface. So you have this filtering element. I was mentioning about missing data, MAF, level of heterozygosity. And this DP for read depth and genotype quality that can be also available, and you can already filter on that. And in terms of groups, you can make up to 10 groups in the system. So to make your discrimination or to make some subset for your SNP marker. And everything is really optimized. So even if you have a very big data file, it works. It works directly. Obviously, if you have a small file, it will be faster, but it can go up to scale with thousands of accessions that are genotype. And associated to the markers, very important, you can insert your metadata. So this is how it looks like. You have a tabulated file that you upload in the system, and it is being converted in this type of interface. And you can filter on any of the columns you have put in your file. In this case, this is a study that we conducted some years ago, a GWAS study on banana. And we put basic element of passport data, the accession name, the DOI for unique identification, which species, which country of origin, which subspecies or subgroup. And based on this, we could do the groups in the interface. So basic passport data, but you can imagine to have more complicated example, like including information on traits. So here I took an example of, again, a GWAS study that was conducted again on banana, but they made a lot of measurement on agronomic characteristic of the plant. And so they could add as many columns they wanted to capture information on traits, like bunch angles, so what were the parents, leaf index. So it's flexible. So it's up to you to select what makes sense to filter. And once you have done this filtering, this exploration, what is very useful is that you can export in multiple formats for subsequent analysis. So you have an example of all the formats that are available with the software. So you import VCF, but you can export VCF, of course, like a subset of your VCF. You can have compressed format to get it smaller. You can convert in AppMap. You can convert it for Flapjack, FASTA format if you want to do phylogenetic analysis. So this is very convenient, again, if you are not a bioinformatician and you don't want to spend a lot of time to converting format. It makes the job for you. And it is modular enough to do it for the whole data set or for groups that you have created or only a few samples that you have selected. And you can decide if you want to export the metadata with the marker or not, which information in the metadata. And as it can create big data set, it's very interesting as well to have an URL to download your file. So you can decide to get this link or to keep the file on server and to share. Either you download your data directly, or you can share this link with someone who will be able also to download the file. So in terms of data use, what can you do? So this type of system, if you import your data early enough during the stage of analysis, can be very useful to work in the gene bank, to look at the diversity, to make core collection, to do some taxonomic identification, gap analysis, look for redundancy in your material. You can also check for genetic integrity. It can be also useful for breeding application, like marker trait association, try to understand what could be interesting donor for specific traits. So all those elements can be done after this exploration, curation, subsetting with this type of tool without the need to be a bioinformatician. And you can do it iteratively. So you can make a first data set, second data set, and so on. So if I try to link with other courses that were made also last week, Damaris, I think it was on Friday, gave some example on how it can be useful to compare different markers. Some markers are more informative than others. And this is the way you can make different subsets, export, and look at the informativeness of your marker, for instance, just as a step, just to really characterize the subset you need for your analysis. And Damaris explained as well the importance of creating core set. So this is what can be supported once you download your data. Genetic structure, Zakaria also made a nice presentation about how you can use SNP marker to look at that, also to do environmental GWAS. So you can export your markers, you can also have your metadata on environment that is in the system. So it can be very convenient to import your data at an early stage in the system. What I described before is when you export your data, but just note that you have also internal applications that can help you with visualization, like Flapjack. So you can directly visualize on the website this type of matrix, where you export your metadata, you export your matrix, and you can benefit from all the features that are provided by Flapjack. Without having to install Flapjack on your computer to convert format again. So that can be useful. And if your data are mapped on a genome of reference, you can look at the context of your SNP markers. So it can be useful to look if your SNP marker are close to a genes directly in the application. We have IGV that is included. And then this is a link with additional website that could be useful for further analysis in genomics, in a genomic context. So if I take an example of rice, you have a lot of material that has been sequenced in the 3000 rice genomes, and other resources were created on Pangenomic to look at the present substance of genes. So let's say that you identify an interesting marker that is in a gene. You could also export the location, go to this website, and look at the distribution of your gene within the sampling of the 3000 rice genome. So it makes a nice link with other tools that are more genomic oriented that then can be useful for breeding, for other type of research. In a similar way, you can link with Garamine, which is also an important resource for cereals, where they keep a lot of, they have, at the moment, they have 28 rice genomes, fully sequenced, annotated, and so on. They also compile a lot of information about markers coming from different studies. So you can, again, link to this type of website and check what are the type of markers that exist in the whole data set beyond your own subset. So that can be an interesting tool for that. So I put a focus on those metadata and variant data because usually this is what is quite difficult to handle, to manage without bioinformatics skills, and I think it can be useful. Then when you want to do data sharing, so one of the last steps of this research life cycle I was talking about, you should consider more than that, obviously. You have to go back to those data that are generated and take action for each category. And each category can be managed differently. And this is what I would like just to touch base now. You have global repositories for data management, and the raw reads of data, what is going out of the sequencing machine, is usually managed by NCBI, ENA. So if you have this type of data set and it is requested, usually when you publish, to put them public, you have to go on NCBI or ANA. You have to create a bioproject, your biosample, to upload your reads. And if you have an assembly, it's also mandatory most of the time to go on Genebanks or RefSeq and to upload your assembly. So if you do it before publication, you have multiple options. You can set embargo dates. It doesn't have to be public now. You can have a link for reviewers, which can be quite handy if they want to check the availability of data. How it looks like in terms of interface. So I took the example of NCBI. This is the one I use, even if ANA is also a very good one. So you create your bioproject, you get an identifier. You can give a title to your project, define the dates, to give a descriptions, a bit more context about your project itself. Then you need to upload information about your biosamples. What type of material was created to create your genotyping data and your genotyping data as well. So for each accession or genotype use, you have to give more information. And this is why, again, it is important to capture metadata early in the process, because if it is not the same person submitting the data at this stage, it can be tricky to get back to the person who collected the data. So this is why you can say what is the country of origin, what is the taxonomy. By the way, the country of origin is now mandatory when you submit your data in those global repositories. And you do the same for your genotyping data, where you have to say what type of machine was used, what type of libraries were used, what are the name of the files, etc., etc. What is the taxonomy coverage? So everything has to be done on those global repository. So it may be a little bit intimidating the first time, but once you get used to it, it can be quite fast to invest. So you can stop here for publication, but I think it's useful practice to go a bit beyond if you really want to promote the use of your data with the community and the traceability. So I will not get back to GIGWA again, but this is a way where you can put your data online and put a link directly in your publication, as we did in some of our research publications. So people can go there and they can interact directly with your variant information. They don't have to download the raw data to process with bioinformatics to predict again SNP that might be different. They can directly interact with your data set, unfiltered or curated. You can put both there and they decide what is useful to them. And for traceability, you have other systems that are quite useful, like Zenodo, Dataverse, where you can upload your file and it's going to give you a unique identifier. So that's I want to represent here. In CGIAR, we use a lot Dataverse. We create a space for a particular study where we upload the file. It can be the variant calling, the VCF, it can be assembly, it can be metadata. And it's going to give you a unique identifier. And this unique identifier can be put as well in publication and can be can be a link, a direct link you give to your partners or collaborators to download the data. They don't have to go back to the publication and so on. So it's a good way to share your data. You can decide about licensing. You can decide if the data are open, restricted or not. So Dataverse is one. Zenodo is another one, which is developed by the European community, which is also a good system to do it. It is well connected, respecting standards like FAIR. So, and if you make a modification in your file because you made a mistake a bit later, you can upload again and you will have a version and you will have your unique identifier for a specific version. And that can be also very handy for traceability of your data set. So with that, just a few take-home message. I hope I convince you that data management is essential. And I know by experience that it is often overlooked. People look after data management at the end of the project. However, it should be done at all stages of the project, starting with the planning, with the metadata curation, until the final files that you are going to share. Extra attention has to be put on metadata because collecting metadata early is much cheaper than fixing later. I like this sentence because this is really true. It can be very time-consuming to get back to people who generated the data. And it can have implication with policy frameworks on DSI and access and benefit sharing. It will be the purpose of the presentation on Friday, this Friday. So if you're interested by that, please connect on Friday. And yeah, so good data management is really key for reproducibility and reuse. That's the end of my presentation. Thank you for your attention. My email address is on the slide if you want to contact me later about it.
Thank you so much, Mathieu. Marie-Noël?
No, go.
Yeah, so since you're getting some applause, yes, well deserved. I will also share mine. So the next presenter unfortunately couldn't be here today. So we will be sharing his presentation as a recorded presentation. So please turn up your volume so we can ensure that you can hear it. I will now share it.
Hello everyone, my name is César Petroli. My background is in genetics and molecular biology. I am a scientist at CIMMYT, working during the last 13 years at GRP based in Mexico. Today I am leader of sequencing and high-throughput genotyping platform at CIMMYT called SAGA, the Genetic Analysis Help for Agriculture. And I am participating in many projects that involves the genetic characterization of gene bank accessions and also breeding materials here at CIMMYT and outside CIMMYT. Then why I want to genotype my gene bank? Well, there are three main reasons. We can optimize the composition of my collection. We can improve the management of my collection, or we can also empower users to identify the best accessions to develop different kind of analysis. The goals for the first one are, for example, represent the full genetic diversity, which I think that it is the more important one. Eliminate redundancy and fill gaps. Stratify or organize my accessions or my collection by the use of gene pools, for example, creating different kind of level, a primary level and secondary level and so on. Between gene banks, we can try to minimize or use the genotypic data to minimize overlaps across those gene banks and being able to create global core collections. For the second reason, we have different goals too. We can confirm the genetic identity during the acquisition and or the regeneration of my collection. Check the genetic drive when regenerating accession of outcrossing crops. Check for some clonal variation during in vitro propagation of accession of clonal crops, and different kind of things to do in terms of the improvement of the management of my collection. And the third reason, we have also different kind of goals or different kind of analysis that we can do using the genotypic data. We can fill gaps in our passport data, very important. We can be enabled to make some genomic selection analysis to try to predict phenotypes. And we can also identify allelic states of key functional genes. Of course, all of these reasons, we will apply different, for all these reasons, we will apply different methods according with my objectives, my goals. For example, for optimization, we will use a method that explores the whole genome to capture the genetic diversity as much as possible. For example, we cannot use a marker panel, you know, to see the diversity of my accessions. Also, for the improvement reason, the cost, we need a method with a very nice cost effective. It had to be cost effective. It has to be a robust data and repeatable between different methods, or between the methods. In the third case, for example, for genomic selection and GWAS analysis, we need to use an appropriate marker density. We can use targeted, like marker panel methods, or make directly the whole genome sequencing. To achieve these goals, the molecular markers have to have some special capabilities. For example, it had to be highly polymorphic. The assay needs to be rapid and easy. The data had to be reproducible between methods or between laboratories that use the same method. If possible, we want to have a codominant marker in terms to recognize the heterozygosity. It had to have a uniform distribution across the genome. The marker needs to be not subject to environmental influence. It has to be neutral, like the SNPs. Of course, inexpensive. We need cheaper markers, or very cheap markers, as much as possible, of course. Easy to be measured, and the data exchange between different laboratories should be very easy. This timeline captures the evolution of the use of marker technologies and sequencing innovations from the 80s to today, 2025, and how they allowed us to improve the plant breeding strategies. We have the basic molecular markers methods on the left side with the RFLP, RAPD, SSRs, some SNPs methods, AFLP. I know that many of you with more than 40 years old, we already use this one, yeah. And maybe the younger people, they don't know how to work with this. But right now, we are moving to from 2011, we moved to GBS technologies and long read sequencing using the third generation sequencers. And now, with a higher accuracy and longer sequences obtained using the fourth generation of sequencers, using, for example, the PAC bioresources. At the bottom, the cost through food diagram, the green area, tell us about the sequencing, how the sequencing has become cheaper and faster, enabling large scale of genotyping and accelerating crops improvement. But also, it is showing in the yellow area how the IT infrastructure needs to be increased to store and handle such a large amount of data. One of the methods that use sequencing to create genotypic data is the DART-seq technology. The DART-seq technology has been created by DART company in Canberra, Australia. And this diagram is showing us the workflow from the genomic DNA to the extraction of two kinds of molecular markers, SilicoDART or PATH and DART-seq SNPs. So the first step, that I think is the more important one, is the reduction of the complexity of the genomic DNA. To do that, we use a combination of two restriction enzymes sensitive to methylation. The common one is the PST1, and the second enzyme will be selected according with the species that we are evaluating. Once we get all these fragments, we add or incorporate a barcode, which is just a little sequence that will allow us to recognize every single fragment from which sample is coming from. Then we add primers in order to amplify those targets. Once we amplify those targets in a PCR machine, we generate a agarose gel to make the QC of the targets and remove those that fail. Once we have that QC done, then we create a pool of targets to create a library and quantify the volume that we will use to be read in the sequencer. So once we get this pool targets in this tube, we this pool includes all the fragments from each sample from many plates. So you can imagine the amount of fragments that we will have here. And then we read those fragments into this sequencer. In this case, at CIMMYT, we have the NovaSeq 6000 from Illumina. And then we use all the fragments to create this library as a base to align then every single fragment against this library, this genomic library. The idea is to recognize the SNPs using this information. After that, using the DART-soft or the DART software, we can extract two kind of markers, the SilicoDART and the DART-seq SNPs. This is a very brief representation about the process, the whole process. So as I mentioned before, we have the genomic DNA, for example, from sample 1 and sample 2. Then we cut the DNA with a combination of two restriction enzymes, and we generate different markers. So the first step is to recognize the presence or the absence of the same fragments for each sample. In this case, the first fragment is present in both samples, so it is a monomorphic marker. The second fragment is present in the first sample and absent in the second sample, so this is a polymorphic markers. And for the third, we have the same marker presence in 2 of the samples, and so on. We can recognize when the fragment is present or not. So polymorphic markers or silico marker will be the one that is present in one sample and absent in another sample. Then this is a dominant marker. The second step is to get all the produced fragments and read it into the sequencer to recognize the nucleotide modifications. This is a codominant marker that everyone knows that call it SNPs. So the DART-soft pipeline allows us to go from the sequencing information to molecular markers, DART-seq SNPs and silico DART. So in this slide, we can see how we go from the light signals from the sequencer coming through the transformation into nucleotide sequence, and then the creation of a simple Excel file that expresses the polymorphisms that make genetically different one sample from another sample. So we report the data, silico DART and DART-seq SNPs, as I mentioned, in an Excel file, a very simple Excel file where the samples are in the columns and the markers are on the rows. The code 1 means the presence of the allele or the marker, and the 0 means the absence of the allele. The white boxes with the dash is a missing data. In the case of the silico DART reports, one row or each row is one marker. In the case of the SNP DART-seq or DART-seq SNPs reports, every 2 rows we have one marker representing one genotype. Also, we have different columns with different QC parameters. For example, the core rate, which is the opposite of the missing data rate. One ratio that is showing us or is telling us about the polymorphisms of each marker. The average of the homozygosity and heterozygosity of each marker, the polymorphic information content of each marker, and also the reproducibility of each marker in the whole population. This parameter give us the more robustness because we just make the replication of almost 20% of the DNA plate, and we use that replicate to see the reproducibility of the data between the original samples and their replicates. And also one of the columns has the sequence information. The main issue in this kind of methodology or technology that use sequencing for create genotypic data is the missing data. Using sequencing, we produce a lot of missing data in terms of course, genotypic data. We have 2 types of missing data, the technical missing data and the biological missing data. For the first one, we have 2 main reasons. The first one is the low coverage in large genomes. That's why it's quite important, if possible, to know about the size of the genomes that we are evaluating. Another reason could be the different coverage caused by the fragment size bias. For the biological missing data, we have 2 main reasons too. The first one could be the genome regions that is not sharing between samples, and the restriction site polymorphisms that it is associated directly with the nature of the enzyme restriction that we are using to cut the genome that we are evaluating. So the missing technical data can be imputed, no problem with that, but the absence of data because of the biological reason cannot be inferred. This technology has some beneficial properties. It is highly polymorphic, creating several tens or even hundreds of thousands of 2 types of molecular markers, silico DART or PABs, present absence of variation, which is a dominant mark, and also the SNPs, the dark-seq SNPs, which is codominant markers. All those markers are reported in an Excel file, very simple Excel file with the code 1, which means the presence of the alleles, the code 0, that means the absence of these alleles, and the markers are identified de novo based on a library, which means that we don't need any genome reference to make the SNP calling. It is important because, for example, for Genebanks, this property allow us to recognize, to really recognize the diversity, the genetic diversity of the samples that we are evaluating. And in terms of sequencing, this method has a large coverage with different options, low, mid, and high density in terms of number of markers that it can produce. For the low density, it produces around 800,000 reads. In terms of mid density, this method, the coverage is 1.5 million of reads, and the high density, the coverage in the high density, the coverage is around 2 million of reads. There is a strict quality control of the markers that DART produce or DART-seq produce. If you remember, I show you different columns on the left side of the Excel file with lot of parameters or quality parameters that allow us to have a very good quality of markers. For example, the call rates, as I show you, the 1 ratio will show us the polymorphism rate of the markers, and the polymorphic information content, the average of heterozygosity, and the reproducibility. The reproducibility is a very important parameter because give us or give to the data a very robust data, comparing the original sample with its own replicates. And of course, in terms of data point, this method is very cost effective. For processes like incrementation of Genebank sections or even for breeding strategies, we have a very large workflow from the seeds to the data or genotypic data production. So in each step, we'll get many different information. For example, in the field books, we have the genealogy information, we have phenotypic data, we have climate data, and we have genotypic data to be associated with every single sample. So it's a very challenge of integrating and tracking a large volume of this large volume of data, even if we, even because we create different IDs for one single sample or one single accession in each step of this workflow. For example, we create one ID for the seeds, one ID for plants from those seeds, one ID for samples, one ID for DNA, one ID for phenotypic data, one ID for genotypic data, and all those Excel files with all that information are in personal computers. So we need a system to combine all those ID and try to minimize the challenge of integration and tracking all that information for each sample, or in this case for Genebanks, for each accession. That's why here at CIMMYT we create a sample tracker system to track each sample from the greenhouse or from the field, going through the DNA extraction till get the genotypic data from each sample or accessions in the case of the Genebank. So the sample tracker system is where all our samples are registered. Then after registered samples, we collect the samples in the greenhouse or in the field, as I mentioned before. With the set, you can see the leaf that we collect here in the left side, and we do this in the same format that we will create the DNA plate for genotyping here on the right side of this slide. So we have all the information of every single accession or sample in the field book. Once we put all this information in the sample tracker system, the sample tracker will create a sample ID linked with the GID, the Genebanks ID, in the format of a DNA plate, as you can see here, 96 wells with 94 samples. Also, the sample tracker system will create a file with all the information that we need to update it or upload, and we upload this file with all the information of every single accession into our DB, our database in the genotyping platform. So you can see here the GID, the Genebanks ID that we got from the field book, and the system create a sample ID linked with that GID. That is the position A1 in the DNA plate, and then we will have that information in the genotyping platform database. Also, the GID linked with the sample ID, so we can track all the information of each accession through the whole workflow. Then, once we have uploaded the input file that we received from the tracker system, our LIMS, the Laboratory Information Management System from SAGA, will allow us to track every single sample from the DNA to the sequence, as you can see in this image. So at SAGA, the Genetic Analysis Service for Agriculture Lab, we use DART-seq technology to create genome profiles of DNA samples. In this slide, you can see the different studies that we have done within CIMMYT and with external collaborators. The left side is showing the characterization we have done with CIMMYT and MACE GenBank, also genotyping of a set of common beans collection from the community of practice in LATAM, in Latin America. I think that you already had a talk about this. In the middle of the slide, you can see the published work of the Mexican coffee and Bromus Uruguayan GenBanks, the famous work of Carolina, of course, with CIMMYT with accessions, and a little work using genotypic data created by SAGA for a wheat GenBank in Azerbaijan. On the right side of the screen, we have using the existing CIMMYT MACE GenBank data to compare with accession from other Mexican MACE GenBank to check germ plants with a seed company, Mexican seed company, and to develop GWAS analysis on BNI trait. This is just to show you the extensive kind of analysis we can do using already existing genotypic data. So as mentioned, at SAGA we use the DART-seq technology to characterize any kind of crops. At the moment, we have done the characterization of almost 50 species, which includes more than 160,000 samples, and maybe more for more than 50 collaborators. So the idea of this platform and, of course, our work is to go beyond of a genotyping service and move to a real research collaboration. So we produce this high quality genotypic data, SNPs and silico darts. As mentioned before, as part of the protocol, we produce 20% of the technical replicates in order to get a very robust data without extra cost, of course. The turnaround time is between 3, 4, maybe 5 weeks. That depends of when we get the samples. We give some support or advice in terms of genotyping design and data interpretation. We can do the blast on different genome reference and consensus maps. We can do the data codes transformation. You saw the reports with ones and zeros, but we can transform the data codes, for example, for HapMap. And we can also include some collaboration on basic diversity analysis. And I think that's
So that was the end of our presentation from César. So César is also online, I can see. So welcome to the call.
Yes, I'm here.
Hi. So would you like to add anything? Otherwise, we will leave space to questions.
No, maybe in the last slide. Sorry, I just connected quite late.
No worries.
Yeah, this is not a good time in the day for me.
Yeah.
Just in the last slide, I maybe forgot to say that we are receiving visitors all the time here at Saga, and people around the world, and from university students from universities, PhD students, and many researchers or scientists are coming from other institutions, CG institutions, and many others national research institutions. So everyone here is welcome to come visit us and learn about what we do here in our lab.
Perfect. Thanks so much. So now we leave a floor to the participants to ask their questions. You can either write in the chat or raise your hand and you will be given the microphone to speak. We have both of our presenters here, so they can respond to your questions. Let's see, I'm checking the chat. Anyone? Questions, concerns? Yeah, we have a hand from Shakil. Please go ahead.
Thank you very much. Excuse me for my late joining. I was busy in another meeting. Probably I might have missed some of the information that I'm going to ask. If that has already been presented, excuse me for that. My question is, for example, if somebody is starting the genotyping or the characterization of the germplasm at large scale these days or recently, for example, so what do you suggest? Which technology would be better to characterize and to develop the DNA fingerprints?
Well, I think that maybe any of the methods that use sequencing could be a good method to characterize or to the discovery phase. When you are in the discovery phase of your research, and you are starting with some new analysis, or if you are looking for the diversity of your materials, depend on the germ plants actually, because if you are talking about some breeding material, could be good to get some or use some methods, specific methods with a couple of maybe thousands of markers producing. So just a couple of thousand of markers or even tens of thousands of markers. But if you are working with some diverse material and you are starting with your work, could be good if you use some methods that produce many markers, and all the markers can cover the whole genome as much as possible, and try to identify that diversity. But always depends of your objective and of course the nature of your germplasm.
Yeah. For example, if someone has breeding lines and then land races, then some of the varieties um, in in such cases, and we have a diverse collection as well. Number of accessions are also higher, and we are starting from the very scratch. Do you think molecular markers like SSRs or any other marker, or DART technology, or whole genome resequencing, or genotyping by sequencing? I mean,
Yeah,
what should be the step that one should take from the scratch?
yeah.
Keeping in
My
view
suggestion, if you don't know about the diversity of your germplasm, you can try first with microsatellites first and see the diversity that you have in your population. And then if you see that you have a lot of diversity, maybe you can try with DART-seq or any other method that produce many markers.
what proportion DART-seq can cover from the genome?
Well, as I said in the presentation, we have different kind of options according with the density that you would like to have in terms of number of molecular markers or SNPs and PAFs. We have three options. The first one is the low density with 800,000 reads. The mid density is 1.5 million of reads, and the high density is 2 million of reads. Of course, with different costs.
Oh, perfect. Thank you so much for
Yeah.
clarification. So if we, yeah, please go ahead.
No. If you need more information, or if you want to have some chat with me or any, contact with me, and you can send an email to me and let's talk.
That is so nice of you. Thank you very much, Daniel. Thank you
You're
so
welcome.
much.
Thank you.
Great. Thank you so much, César, for taking that question. We have another question in the chat from Carmen. Is the sample tracker system available to all users or only for CGIAR scientists?
Yeah, in this case, it was for every CGIAR scientist. We are using that system here at CIMMYT. I believe, I'm not sure, that is public for anyone, but maybe you can contact me again, and I will just readdress your email for the responsible for that.
Noted. I will put your email in the chat so that everyone
good.
can speak and send you their questions.
Maybe,
So,
Alessandra, on this question, I can complement a little bit, because
please.
I would say that the sample tracking system is even not shared within CGIAR. It's specific to centers. And there is a plan maybe to develop some extension like that in GGC, Green Global Community Edition. That is a system for data management in Genebanks. So many people are missing this type of feature, and many centers have tried to develop with their own needs. So makes sense to maybe at some point to have something generic for all.
Yeah, you're right, Matthew. Yeah, in terms of the sample tracker, I believe the capability of this system was quite small, not that big, to support the entire CG system. Here at CIMMYT, it's working quite well, but you're right. I think that people is thinking to move to another system in order to have more capacity. We are talking about thousands and thousands of samples, and it's quite difficult to manage all that kind of information.
Thank you, Carmen. I hope that answers your question. Are there any other questions, whether in the chat or you can raise your hand? Yeah, Elizabeth, please.
Good morning. Good morning to all. Good afternoon, good night, wherever you are, colleagues. Thank you so much, Matthew and César, for this great presentations. I mean, it's really mind-boggling where the technology is at this point. Is it possible for a breeder outside of the CGIAR system to work with you to help with in identifying or selecting marker-assisted selection of their populations? For example, a rice breeder in Guyana, for example, to help identify some of their aromatic progeny from crosses. Is that possible to work with the SAGA system?
Yeah, sure. In terms of SAGA, of course, you can work with us. Actually, this is a service. We have already data from our Genebanks here, maize and wheat. The genotypic data of those banks are public. So you can even use the genotypic data created for the Genebank. That include all the accessions that are in the Genebanks, including lines and hives.
But this is public. Rice.
Yeah. Oh, sorry. Well, that's what we have here as a public genotypic data. But of course, you can contact us, and we are working with people in rice, and we have some data. Of course, we have to manage how to share the data if these people from Embrapa, for example, we have some data from the Embrapa Genebank of rice, and maybe if you want, I can ask them and see if we can share data with you. Maybe you can genotype some data with us, and combine the data and compare the genetic background of your material and Embrapa and any other research institutions that have genotyping with us. That's possible.
Okay, Daniel. We'll reach out to you. Thank you so much.
Okay, welcome.
Thank you, Elizabeth, and colleagues for responding. There's a question on the chat that is asking, how is the tracking system useful for benefit sharing of genetic resources? I don't know if any of you can answer this question.
Yeah, I mean, it's not the tracking system that has been presented. And César, sorry if I take the question, but is more for a technical perspective, is to make sure that you link your physical sample from the Genebank, for instance, until the data generation. But it's not really a tracking system that you use for checking who is going to use your data. And so maybe this question is referring to the discussion that happened in CBD and Plant Treaty about maybe tracking the material which is used and with generating DSI on the material. But it's a different system. And so if it is related to DSI question, again on Friday, there will be an interesting perspective on policy about access and benefit sharing.
Yeah, that's exactly it. So if you have any more questions regarding the benefit sharing system on Friday, I think you will get a lot of interesting perspectives on that from our colleagues in the policy team. I see a question from Knan Dalem. In the previous presentation, we have seen a criteria for quality control. Is it fixed or flexible depending on the objectives we drafted? Why are those criteria ideal?
I think it is referring to one of my slides on filtering, if I'm correct, where there is I was showing the format of the VCF, and then that using GIGWA you can also give a threshold for filtering. Well, you have usual threshold that are put in publication, so you can use variation in the threshold, but this is exactly why you have this possibility. But like rip depth, it will depend on your organism and what you want to do with some analysis. In our case, we work with polyploids, So if we want to have enough coverage by haplotype in our polyploids, we usually select with a read depth that is a bit more important than people would do for rice. Yeah. So this is the type of element that you can fine tune in the criteria. But for the quality, I mean, the quality itself, the threat score is usually quite standard, and probably you deal with that assessment.
Yep.
To repeat the question, yeah.
Yes, about the QC, I'm not sure if the question is exactly what I understood, but in terms of QC and what are the thresholds that we use in terms of the market quality, that depends also of the population that we are evaluating, the germ plants that we are evaluating. If we have breeding material, we are more strict in terms, for example, of missing data. We try to avoid and delete and not report all those markers that have more than 20% of missing data. But if we are working with some diversity or very diversity material, we are more flexible and relaxed and try to include more markers with a bit more of missing data, maybe 40%, 45%, because I believe that maybe we are missing information over there. So basically the user can, let's say, play and try to build her own or his own concept about where is the threshold that we have to use. And of course, you have a lot of papers talking about. You can search.
Perfect. Thank you so much, César. The colleague can let us know if that answers his question. I see there's another question from our colleague Martsikwan from Lesotho. The question is, sometimes the results of genotyping data show scaffold, where it's supposed to show the chromosome position. What is the meaning of scaffold?
I can take this one.
You know, please.
Sorry. Yeah, it's when your data are mapped on a genome of reference. So usually your data are mapped indeed on chromosome, but it really depends on the quality of your genome of reference. So sometimes you have some scaffold that could not be included in chromosome. So if you look at the full genome assembly, you will have your chromosomes, and at the end of the file, you have a number of scaffolds that could not be placed in your assembly. So it just means that your reads were mapped on the sequence, and we don't know exactly on which chromosome they belong. So yeah, so it doesn't give you the exact position on chromosome, but it corresponds to somewhere in the genome. It's just that you don't know exactly where.
Yeah, I agree with Matthew. Yeah, especially because you have, that's why you have, for example, many different genome reference for one species, you know, because people have different information or sequences to build the genome reference, and sometimes your sequence, specific sequence, are not part of those chromosomes.
It can be also an indicator of the quality of the genome that was used for the mapping, because we have less and less type of case, because the most recent genomes that are sequenced are only chromosome scale, so you don't have much more scaffold anymore. So if you're in your result, you see a lot of mapping on scaffolds, it can tell you that the genome used for the mapping may not be a very, very good quality. It's maybe a draft genome.
Yeah, and people is updating the reference all the time. So maybe in a future, your sequence is part of one of the chromosomes.
Thank you, colleagues. I see Shaquille, you have your hand up again, so feel free to speak whenever you want.
Yeah, thank you very much. My question is how to cope with that challenge if the genotyping results and the phenotyping results are contradicting with each other for a same accession. As the sequencing is progressing very rapidly, most of the gene banks and plant scientists, okay, they are characterizing their germplasm using sequencing technologies, either by whole genome resequencing or genotyping by sequencing or any other partial genome sequencing technologies. In some of the cases, we can see that our genotyping results cannot be the same as our phenotyping results if we have done the phenotyping according to the UPOV characteristics. So in such situation, what do you suggest? For example, I'm facing such type of issue here during my job. Some of the accessions, they looks like they are, I mean, they are similar according to UPOV characteristics, but when I genotype them using whole genome resequencing at the depth of 10x, they showed differences according to their sequencing data. So in such situation, what do you suggest where we should go, which result we should consider as an appropriate or the solid result? Thank you very much.
Yeah, and in that case, I believe that if you are focused on one specific trait, maybe you can build some marker panel. For example, Dart has the Dart tag method. They have specific panels with 2,000, 3,000 of markers that are associated some way with that specific trait, and then you can just test your population. If your population have that markers, that's possible that the chance to get the that trait is higher. But the first step, maybe you can build some kind of marker panel or design a marker panel for those markers that are associated with your trait, and then we just or the company or the technology that you use will just build this marker panel, and then just check if your population has those markers.
And probably also important to discard that it's not a sampling issue or mislabeling issue, because sometimes you can have some plant in the field that are not the same that were sent for sequencing. And so a double check can be useful, because it's just that you don't have exactly the same sample. And it happens from time to time in Genebanks as well. And that's why it's quite useful to genotype.
Yeah. Sometimes we have a very nice Excel file with all the data, saying that we are in front of the correct material or samples, and 1, 2, or 3 of them are very different, and the people just track those samples, and they're not what they expecting to be.
Perfect. Thank you very much. Another linked question is, while curating the gene banks or the germplasm, if we have found a lot of duplications, what do you suggest what to do with those duplications? Of course, we are not going to discard them. If we are still keeping them, then why we are curating the gene banks? Why we are curating the germplasm?
Matthew,
my
answer was to discard them.
No, well, maybe not.
I'm not gene bank guy. So.
Well, again, it depends a lot about what type of crops. For instance, for clonal crops, there is a high cost to keep the clonal crop in vitro. So putting them in cryo is a way, to conserve them without trashing them, just in case. You never know. And for clonal crops, what is really a duplicate? Even sometimes you sequence the same sample with full sequence, you compare them and you have variation. So it's sometimes difficult to decide what is really a duplicate, or if you can do it with passport data, it's another question. But I think if it's not too costly, like seeds, and you keep it in a box, just keep it long term, and you don't put it in your distribution material. That is probably taking a bit more effort to maintain. Clonal crops, cryo. And yeah, that's why it's important to characterize, because you can decrease the cost of operation for the materials that you distribute a lot. And then if it's not too costly to conserve like cryo or long-term for seeds, I mean, why should we trash them? We never know. Maybe it may be useful in the future.
Absolutely. Yes, perfect.
Yeah, agree.
Thank you very much.
Thank you, colleagues and Ahmad, for that very insightful question. I saw there was a question from our colleague from Mali, but this was already answered in the chat by Marie-Noël, so I will skip that one. Perhaps I will take one final question since we are already over time. From Fernanda, she's asking, at our institution, we perform microsatellite markers. How far behind are we from all the countries that are doing SNPs? And when we can have this technology, can we keep the information generated by microsatellites, or is it better to start with all the crops that we have the data of microsatellites?
No, it is possible to combine different kind of genotypic data. We can combine microsatellites, for example, with microarrays. We can combine with the PAPS data that we produce. But of course, if you want to start with the bigger characterization, you should use some methodology or technology like DART-seq and try to cover or represent the diversity of your material as much as possible. But of course, we can combine data or try to combine data to make the microsatellites even useful.
Mathieu, do you want to add anything?
Yeah, I was reading the question, how far behind are we from all the countries that are doing SNP? It's a difficult question to reply, but I think it's also a matter of scale and cost, because sometimes doing microsatellites on a large number of samples can be as costly as doing SNP now. So it will depend also if it is a quick verification for a few samples or exactly. If you can, yeah, if you plan carefully, maybe you will have the same cost, and you will have the advantage to maybe link with genomic data if you see genome of reference, so and the type of use. So it's a bit case by case. Yeah.
If you want to make some paternity tests, just use your microsatellites. Don't jump to a very big technology. If you want to do your QC, use, for example, DART-LD or CASP. You can use cheaper methods to make your the QC of your incrementation, for example, your Genebanks incrementation. But that depends on the objectives of your work.
And the infrastructure that you have, because even if you have the same cost, I mean, the data processing would be a bit more difficult for SNP Absolutely. Data, because you have all the storage of data, as I mentioned, the bioinformatics processing. So if your center is equipped with that, it's fine. If it's not, obviously it's going to be a bit more difficult. It requires some collaboration, some investment. So all those criteria to be considered before putting in place the experiment, I would say.
Thanks so much, colleagues. If that's all, I don't see any other hands up or comments. So Marie-Noël, would you like to close this call, and I can help you remind the colleagues of our meeting tomorrow, which will be at the same time as today, 1 p.m. Central European Summer Time.
Thank you very much, Alexandra. Thank you very much to our speaker for the excellent presentation, and to all of you for your active participation in today's webinar again. So on behalf of the organizing team, I would like really sincerely to thank you, and we look forward to see you tomorrow again. As Alexandra said, please be on time. Tomorrow, 1 p.m. European time, and Friday will be 1:30 p.m. European time. Thank you all. Bye-bye.
Bye.
Thank you so much.
Thank
Thank
you.
you.
Bye-bye.
Thank you.
Thank you.
Bye-bye.
Take care.
Thank
Bye-bye.
you.